I will try to tell you a little bit about our experience with annotating targets in the context of diseases and drugs, and perhaps try to address the issue, can we use informatics to improve on therapeutics? Most of you might know that we are here because we're funded through the same team, which is an NIH Common Funds initiative called Illuminating the Genome. We are working in the Knowledge Management Center, which includes teams from Denmark, England, Miami, NIH, and Mount Sinai, in addition to UNM. So this is a partial list of the collaborators that are working on this project. What we are aiming to do is essentially try to use automated, semi-automated, and human curation processing for a variety of data. And in reality, the data is so complex, I'm quite sure that we will not do it justice in the 2 years for which this program is funded. But essentially, we have to try to capture as much data as possible. For example, Campbell that John presented, as well as Shore Campbell, are part of this automated process system. And then we are developing, as we speak, drug target ontologies for not me in particular, but Stefan Schurer, as well as Lars Jensen, are working on this. We try to plug in with a system called disease ontology, which is a separate, not related to us, NIH-funded project. I will mention that one more a bit later. And then, of course, we try to include as much data from literature, clinical trials, drug labels as possible. And when all this is assembled, it ends up into Target Central. And, NIH NCATS is developing a user interface portal to help with mining the system for the outside world. And the way we envision this, we also have expert target panels. So, for example, Brian Roth from the University of North Carolina will lead the GPCR target panel, and then Michael Vith from Lilly will lead the kinase panel. And so we will have additional human curation to support the prioritization process. It's a really complicated process. We are trying to make this as integrated as possible. Two very interesting developments from the Novo Nordisk Foundation Center for Protein Research are diseases and tissues. These are developed by Lars Jensen. Campbell and Shore Campbell have already been mentioned. At UNM, we have something similar to Campbell, which we call CARSPAT. It includes additional databases such as Brian Roth's PDSP and the UFAR's Guide to Pharmacology, and another database called UMBAT. And then at Mount Sinai, the enricher system is being developed. I don't think this audience needs introduction to that. And then separately, we interface with Antibodypedia and with the disease ontology system. And these will all become available through Pharos over time. And pharos.nih.gov will be the designation for this system. We are interested in prioritizing targets. It turns out there is not a single simple definition of target. The most we can agree on is that it has to have a molecular mass and it's a material entity. So a lot of people seem to consider that the drug target could be a pathway. A pathway is a concept which depends on who you're talking to and whether you consider that pathway to be relevant for cancer, for a pathological process, or a normal process. And in particular, if you perturb the pathway will rearrange itself and it will become something else. So we sort of assume that a drug target needs to interact with the drug. So that's why it's a target. And then its manipulation will lead to something relevant in the clinic. So within that context, we try to produce this sort of definition so we understand what we're working with. So part of the IDG proposal, we came up with this target development level, which currently has 6 layers. T-clean plus is currently the most relevant category in the sense that it associates proteins that we know bind to drugs, and it also associates to mode of action. So those are the creme de la creme of drug targets. But in a simpler definition, T-clean are proteins that we know interact physically with drugs with an affinity better than 1 micromolar. We had to relax that criterion to 10 micromolar. For ion channels, they tend to be weaker in potency. If you look overall at drugs that are annotated with high activities. T-chem are pretty much the same as T-clean, except the chemicals that interact with them are not launched drugs. For T-macro, we lack any knowledge of small molecule chemicals that bind to these proteins, but we have a wide variety of associations to diseases and wide representation in literature that allows us to characterize the function of these proteins. There is a more limited degree of information available for T-gray, and then T-dark is basically the set where we know nothing about. So, as it looks in the December version of IDG, the target development level annotation, for the 4 target families that are of interest IDG, and this is a complete list of the human proteins that we track. We roughly have 332 in T-clean plus, 330 in T-clean, and then about 100 in T-chem. And then as you progress to T-macro and T-gray, you can see the numbers. Some of the criteria that we use to annotate T-gray and T-dark, in addition to the lack of disease annotation, we put a 90th percentile upper limit for reference into function. These gene riffs had been suggested by Andre Goulart, who works with Icahn. And then we're also looking at a literature score that comes from automated text mining from Lars Jensen in Denmark. We initially had PubMed scores, which were just a one-to-one association, so anytime a protein would occur in a paper, we would give it a score of 1. But Lars has developed a rather sophisticated system to score proteins, where a protein that occurs only once in a paper, but 100 other proteins occur in the same paper, that gets a fraction of a score. So that's why you have non-integers in that. And then Matthias Gulen at Karolinska has started this antibodypedia project. We interface through an API and extract both the total count of antibodies as well as just monoclonal antibodies for each protein. So we count antibodies, and then if it's under 38, that would be a T-dark protein. And you'd be surprised how many antibodies are actually available for certain proteins. So if we try to answer the same question for the entire proteome, regardless of target family, these are roughly the numbers. If we keep track of disease annotation, and this comes from the text mining system developed by Lars Jensen in Denmark, there's about 700 proteins in T-dark that have somewhat significant disease annotations, and then 800 in T-gray, and then 4,000 in T-macro. And then again, if you look at the reference function, those are the numbers that pan out. So roughly 10,000 proteins, so about 50%, more than 50% are in the not illuminated part of the genome, so most of the work is still ahead of us. If you were to ask the question from a drug discovery perspective, it turns out that more than 60% of the folks' druggable targets lack therapeutic agents. And so that refers to this, which these are the druggable targets. And then if you look at the entire human proteome, it's even worse. It's more closer to 90% of them that lack chemical agents. So from a medicinal chemist's point of view, the field is wide open. There's a lot of work to be done. One of the tools that we've used to work in this area is a database that we call DrugsDB. We used DrugBank and other resources just like everyone else until we realized that we always came to the problem of mapping drug labels to what we call APIs, which is active pharmaceutical ingredients. Having the definition of a drug depends whether you talk to a pharmacist or to a chemist. And yes, in the UK, a chemist and a pharmacist are the same thing. But if you talk to a physician or a pharmacist, a drug is a pill. To a chemist, a drug is a molecular structure. So we had to develop a tool that would actually map drug labels onto these active pharmaceutical ingredients. So we currently track more than 52,000 approved drug labels. So those come from FDA, and we access them via the Daily Med system. We then map them with technologies that are compliant with the Unified Medical Language System and this 08 Odyssey technology. And then we track these number of chemicals, these number of biologics. We map them into RxNorm. RxNorm is maintained by the National Library of Medicine. It's used by most hospitals, which go back from RxNorm to the national drug codes. So any clinic will rely on those, not on chemical names. We also maintain mapping to ATC, this Anatomic Therapeutic Classification System, as well as the international non-proprietary names. Both of these are of interest to the World Health Organization. So WHO has an interest in these systems. And then this is the number as of last night of chemicals that map all RxNorm, ATC, and INN. So that's probably the most, like the super drugs, if you want, because they're of interest across the globe. And then this is the number of targets that we track for these drugs, as well as numeric activities and mechanism of action. So the mechanism of action, I'll tell you about that in a second, but I thought that would be the segue. This is actually an interesting problem, capitalism in the pharmacy. There's a huge gap, roughly 5 to 1, between over-the-counter medicines and prescription drugs. The prescription drugs, of course, include on-patent drugs as well as controlled substances. But if you look at the over-the-counter medicines, and I have to say I've been doing this slide so I can track it all the way back to 2010, and this number just increases and increases much more rapidly than this number. It used to be like 18,000 to about 18,000, now it's 28 to 23,000. And I call this capitalism in the pharmacy. There is more than 3,000 packages that contain acetaminophen. It's just mind-boggling. I mean, between CVS and Walgreens and all these vendors of drugs, they come up with their own system. And then there's, for example, Tylenol for menstrual relief that costs about 3 times as the generic, and it's the same ingredients. Yeah, so they really prey on the unsuspecting persons, and it's there's also little remedies. Little remedies cost more than, because that's for babies. So, yeah. So, back to modes of action. We are working, have been working with John for, or independently, I should say, for several years to try to annotate the mode of action for drugs. And a lot of people will start their work by saying, I work on GPCRs because GPCRs are the most important target class. 50% of drugs bind to GPCRs. That's an urban legend. Maybe 25% of drugs bind to GPCRs. If you look, so we track kinases separately because of IDG, but these are enzymes and these are kinases, so they should actually be in the same bin. About 33% are enzymes, and then ion channels are a close second to GPCRs. And then there's nuclear receptors and transporters that are of interest in general terms to modes of action. This is a separate subset where we do track the numeric, so numeric annotation, and in that case, because of promiscuity of what's called Class A amine GPCRs, there are a lot of drugs that hit Class A GPCRs, and in particular about 40 of those. And so there is a net enrichment in bioactivities when you look at the GPCRs, also causes of side effects, as you can probably imagine. And so this is currently where we are. We have about 700 FDA-approved APIs that we map back to 1,300 numeric annotations. Most of them are human, some of them are non-human, and of course the non-human reflects antibacterial activities, but the human ones reflect the multiplicity of targets that are typical for drugs. And then on the top targets, we have things like the glucocorticoid receptor, histamine H1 receptor, GABA A. This is your typical benzodiazepines, sodium channels, etc., etc. So I also wanted to go from the general to the particular to illustrate why this is not a trivial process. And the years of manual curation have gone into this analysis, which, John, is there a manuscript that's supposed to go out soon? Or silence? Okay. So, for example, we try to identify the influence of transporters on the clinical activities for the histamine H1 receptor and the alpha A1 adrenergic receptor. And both of these are associated, so when you bind to that receptor in the brain, you can associate with a severe side effect, sedation in the case of H1, and orthostatic hypotension in the case of alpha-1A. The reason the drug does not have that side effect does not relate to binding or not binding, which is of course a prerequisite, but to the fact of how long it stays in the brain. So if you look into medicinal chemistry literature, they will tell you that when they optimize for what they call second generation antihistamines, they change the distribution of the drug so it doesn't go into the brain. That is an urban legend. And in this paper, we argue why that is. The short story is that if your drug, and these are second generation antihistamines, if your drug is a substrate to a pump called PGP or ABCB1, it depends on who you talk to, it gets effluxed from the brain. It basically goes into the brain, but it doesn't stay there. And because it doesn't stay there, regardless of the dosage, it will not have a side effect. We do see that the dosage has an effect. So those that have a severe side effect, and this is also regardless of the potency to the receptor, it does, those that are not effluxed have that side effect. So that sort of diagonally you should look at the way these are separate. So we applied what we call a Popperian hypothesis. We tried to falsify this, and the way to falsify this was to change completely the receptor and the biological effect. So we look now at orthostatic hypotension in relationship to alpha-1A adrenergic receptor, as opposed to the earlier slide that was histamine H1 versus sedation. We basically get the same image. I mean, you'll think the image is the same, but it's not. And the numbers are slightly different. These are independently annotated drugs. The short of it is, if it doesn't go into the brain, it will not cause a side effect. So the publishers requested that we put this in an image because they wanted to put it on a journal cover. So the short of it is that you have a drug that goes into the brain. If it goes into the brain, this in particular would be your typical anti-allergic drug. Its target should be the histamine H1 receptor in the periphery, not in the brain. If it goes into the brain, it causes sedation, sleepiness, drowsiness, whatever. By the same token, in this case, we argue if the drug doesn't go into the brain and it's a histamine H1 antagonist, such as the newer ones like levocetirizine and loratadine, it gets efflux from the brain. So we consider both the peripheric histamine H1 receptor as well as the ABCB1 pump as targets. So ABCB1 in the brain is a drug target for the antihistamine drugs, because without it, they would be less effective. And in fact, what you see is that if you overdose even the second generation antihistamines, you saturate the pump and you will still get the side effect. So you pretty much get the same. So the conclusion is that the transporters mediate CNS side effects. And that's why non-sedating are functioning. And then, of course, we all know that in cancer, the same pump ABCB1 is an anti-target because it effluxes the cancer drug from the cell, and that's by itself not a desirable interaction. So how do we try to keep track of this at the global level? We are also asking the question, how many diseases do we monitor? And so in this column, or in this row here, we're looking at unique disease concepts that exist on label. That's about 2,000. And these are unique drugs. So this is all the drugs that we currently track, contraindications, mechanism of action, and then off-label. And so we're essentially trying to ask the question, how many diseases do we address? And there is a website, disease-ontology.org or -ontology.org. And if you go to that website, you will find that there are about 8,000 diseases that are currently being annotated. And of those, we track about 2,500 with both on-label and off-label indications. So roughly 70% of diseases lack therapeutic agents. Granted, some of them have a surgical solution, but not all. So again, we have a really wide open therapeutic application, or the realm of applications is wide open. So for the next 10 minutes, I'm going to share some of the my thoughts on drug repurposing from an academic perspective. And there is now a journal called the Journal of Drug Repositioning, Repurposing, and Rescuing. And we also, the reason we are here today and tomorrow is to talk about computational ways to address this as part of the IDG. So before I tell you about our experience, I want to give you an overview of the impact of layoffs from the pharma industry on patents. So if you look, starting with 2003, 2004, the number of patents that are awarded for drug discovery in academic centers has pretty much doubled. over the space of a decade. And if you compare that with what's going on in the industry, you can see that there is a net trend that's going up and up, which indicates that academic patents are on the rise. This slide was generated about 3 years ago. It was an idea that came from Chris Lipinski, one of the most recognized household names in medicinal chemistry. He worked for nearly 4 decades at Pfizer. So we came up with the question, well, how many? We didn't publish this because we disagree on the interpretation. I suggested we do it as a Socratic dialogue where I put my comments in one way and Chris would put his views in another way, but it didn't amount to anything. Chris Lipinski argues that patents from academia are not worth anything and they're just polluting the patent field. I disagree, so I'll let you make your own interpretations as whether this is true or not. And maybe because you're academics, you're biased. So, if you were to design a drug repurposing workflow, this would be a typical scenario. You start with a library of drugs, whether it's virtual or physical. You run either a physical screen or a virtual screen, and then you come up with some new targets. Unfortunately, for publication purposes, most people stop here. They come up with a new target activity, and then they say, hey, this is where I'm interested, I'm going to publish this, and the rest doesn't matter to me. It's quite important, I would stress, that you should confirm this in the clinic, so there should be some clinical trials after which you can talk about the successful repositioning or repurposing. This is just an example. It irked me at the time because I was, of course, interested in MRSA. So you all know about this methicillin-resistant Staph aureus. This is a news outlet, BBC, which published this particular story. Vitamin B3 helps kill superbugs. Okay. Well, it turns out that if you actually read the paper, nowhere in the paper MRSA is mentioned. As far as I know, MRSA is a different strain than your typical Staph aureus, so you shouldn't actually make a claim unless you test it. But if you read the fine print, it turns out that the way they've done the experiments, you would have to take about 4 times the maximum recommended dose of vitamin B3 in order to get any effect. And it's not likely that you're going to do that because you cannot tolerate that kind of dosage. I mean, they tried it with patients in cancer, so I really looked at the highest dose ever given in man, but at doses above 85 mg per kg, your kidneys begin to stop. So you can't really bathe your blood in vitamin B3 so that you kill Staph aureus. So, in other words, it doesn't work. But what's more annoying is that they actually sold it as a different story than it really was. Okay? And then, of course, patients say, oh yeah, there's hope, we're going to get the cure. So how many times this happens? Quite often. So the way I phrase it, it's generally believed that you need to do fewer clinical studies when you do drug repurposing. I've been doing this for almost 10 years. I have come to the conclusion that's not really true. I think you can quickly reach 10 million minimum that you will require to do drug repurposing for 2 reasons, efficacy and safety. You still need to do clinical trials for both. And most likely, people in the pharma companies, they're not really idiots. They will look for clinical effects, and they will look for targets before they come up with the patent and before they put the drug on the market. So once the drug is approved, they will probably do the repositioning themselves. Some of you might know that Pfizer has Viagra approved for erectile dysfunction. The same drug, but at 1/3 of the dosage, so it's formulated as 20 mg tablet as opposed to 60 mg, is called Revatio, and it's approved for pulmonary hypertension. So if you have pulmonary hypertension and you take 3 times the dosage, then you can have a maritime. Okay? via the Clinical Translational Science Network, or in Europe via what's called the Innovative Medicines Initiative, there are a number of industry academic partnerships that focus on trying to bridge this valley of death. And that expression, by the way, made its way in the congressional record. So you can look it up. And it's all related to drug discovery. This was the law that supposedly was going to give $500 million for the Cure Acceleration Network. Of course, they all voted it, but they didn't appropriate the budget. So $500 million quickly turned into less than 10 million, as far as I know. But that's politics. So I'm going to skip the rest of this slide. From our perspective at UNM, we've worked on a number of projects. The one that I found the most intriguing was that you take a typical nasal steroid, and it turns out it's a really good inhibitor for ABCB1. And we were thinking that, well, if you block ABCB1, then your typical cancer drug could do its job. At the time, we were all excited to pursue clinical trials with this. One of our colleagues, Rob Cromas, who we were working with him on a different project, he published a paper in Blood with the title, Time to Dismount the MDR Horse. MDR is another name for PGP or ABCB1. And dismounting a horse is in Indian, the way Native Americans talk about it, you dismount the horse when the horse is dead. Okay, so there was really good evidence that you should not pursue blocking ABCD1 as an adjuvant target in cancer. We did have some success with Ketorolac, so I'll show one slide for each, Repurposing and Ketorolac. And we are also quite able to just do applied virtual screening and then try to identify targets for toxicity. And, this one we published in Clinical Pharmacology and Therapeutics, but what was good for our case was an accompanying feature that was co-authored by 5 people at the FDA who were quite happy that we can use cheminformatics to try to annotate side effects. So, raltegravir, the story there was that we were having a discussion with Rob Cromas, who was at the time working on metnase, and then it turns out that metnase has a transposase domain. And so what we did was to procure raltegravir from China at $800 a milligram, and we essentially had it tested. And then it turns out that when you combine it with etoposide, so etoposide itself has no effect on this etoposide resistant or whatever they're called cell line. But when you combine it with raltegravir, it actually allows etoposide to do the DNA damage. So the way metnase works is it's a DNA repair enzyme. So as you try to damage DNA with etoposide, metnase will work to fix it. If you inhibit DNA repair, then you can enable DNA damage, and then you slow the growth. A clinical trial was conducted for about 2 years at UNM. It was stopped primarily because at the maximum recommended FDA-approved dose, patients did not tolerate raltegravir. And I have since found out that if you actually look at the literature, it's used for AIDS patients, but those patients are gradually exposed to raltegravir until they tolerate those levels. And so, in my opinion, I think when the trial was put together, they did not take that into account. So if you just take patients and simply give them high dose of raltegravir, it doesn't work. So with Ketorolac, we have a slightly more intriguing story. Initially, the drug that we identified was arnaproxen. This was work done by Angela Ness. We conducted a high throughput screen. Naproxen is part of the Creswick library, and then it turns out that even in cancers, it actually works effectively to block xenografts. In xenograft models, it's effective as an anti-cancer drug. So I applied simple cheminformatics. I initially suggested a related compound called nabumetone. Nabumetone is very similar to naproxen, except it lacks a metal group, which would probably be somewhere there. So it's what I call an alpha metal group compared to the carboxylate. It turns out nabumetone is inactive. And so that led to Ketorolac, because Ketorolac is approved as a racemic drug. So RS Ketorolac is given in humans. And you can see here at the in the same model as R naproxen that R Ketorolac is more effective than S Ketorolac. And, there is a clinical trial that's ongoing at UNM. About 20 patients have been successfully exposed to oral Ketorolac, stage 3 ovarian cancer. They are collecting ascites fluid and measuring R Ketorolac, which turns to bioaccumulate preferentially. So after 24 hours, you get only R Ketorolac remaining in the system, and the concentration of it in ascites fluid is enough to block the targets, which are RAC1 and CDC4. So it's quite an exciting story.
Was that a direct effect or secondary to cyclooxygenase inhibition? So
everybody asks the same question. R Ketorolac has no effect on, or very little effect on cyclooxygenases.
Sure, well.
Yeah, I'm sure. And if you have additional data that suggests otherwise, I'll be happy to look at it.
But the literature, that's not. There is data showing that anti-inflammatories or aspirin also reduce cancer.
Yes, I'm aware of that body of work. Yeah. Well, we actually think it has to do more with this. So, what we want to do with respect to drug repurposing is to propose to the scientific community that a simple classification scheme should be used to classify this project. So my favorite is there are, I think, every month more and more papers published in journals such as PLOS Computational Biology that completely lack any experimental evidence. So I would call that level 0. And then level 4, of course, it's on label, it's well documented, etc. And then in between there are degrees of scientific evidence. One's in vitro, second one is animal studies, and then the third one is proof of concept. And what we also have difficulty in addressing is how obvious it is to repurpose a drug. So when somebody goes from oral gingivitis to oral mucositis, there is such a project. We call that trivial. When you take thalidomide and you repurpose it for myeloma, whereas initially it was 50 years ago for, pregnant women that had difficulty with their pregnancy, that would be a very different repurposing project. So the difficulty level is quite high. So just to give you some examples from our own group, we had a case where we studied benzbromarone, which is a drug used for gout, and it really showed promise. So in vitro, it's a wonderful blocker of the receptor for quorum sensing in Staph aureus, but it doesn't work in vivo. We took a small molecule that's similar to benzbromarone, and that works in mice. So we're pursuing that, but benzbromarone itself, it's a closed story. We have some activity for Astemizole, but we do not follow that up, mostly because of funding issues. And then with Ketorolac, we do have, we show effectiveness in stage 3 ovarian cancer. And then, as I mentioned, Rebatio versus Viagra, and then thalidomide. You can also go to complication of leprosy. So those are quite well-known studies. So is there a database for drug repurposing? The short answer is there is no resource with reliable information. We found this website called drugrepurposing.info. It only has protected access. Every time I try to access it, it will require money, so I couldn't do it. But there is also a free site. If you look at it, there are 63 entries, 44 small molecules, 5 biologics, and then there is this very interesting molecule called tartiramere acetate, which is a non-biologic complex drug. Yes?
I've got 5 minutes.
5 more minutes? Oh, then I'm really in trouble because I have lots of more slides. Okay, so we're trying to address the question, can we do repurposing if it's off-label? So now I guess I'm going to... the real challenge in this case is how can you move drug repurposing projects into the clinic? And we try to follow this up quite often. For example, there is a project which suggests that you could use orlistat for malaria. John, was this in Nature Medicine or was it a science paper?
Science.
Science. So orlistat has zero bioavailability in man. Okay, its purpose is to block a lipase from the pancreas, so it blocks lipid absorption from the gut. It has zero bioavailability. How are you going to repurpose that for malaria? I have no idea. The other one from the same paper was dextroamphetamine. You'll get them high before you block malaria. So, and that's just one example, and there's many of those. So I'm going to have to go very quickly and skip a lot of this. If you have questions about this, I'll be happy to discuss. We have ways to classify targets. So one example I'm going to show right now is, recall that we have about 600 targets that are classified as T-clean plus, meaning they're associated with mechanism of action. And then if you look at the entire proteome, just at high sequence fidelity, we have about 32 targets that we prioritized. And those come from clustering with similarity and T-clean plus, as well as strong disease associations. So this is a list of targets that are potentially druggable from the IDG initiative. These are GPCRs, nuclear receptors, ion channels, and then kinases. And then out of this list, we manually curated even strong associations to PubMed. So we have some targets which could potentially become druggable just based on the strength of their prioritization as well as the diseases that might be relevant. So we don't know the answer to that, of course. IDG also collaborates with the International Mouse Phenotype Consortium. So about 2 years ago, NIH gave a list of 300 such genes to IMDC. And then it turns out that if you map them to our own prioritization system, about 60% of them fall into the category that we don't know much about, so between T-dark and T-macro. And there is a website, so IDG has a landing page. The link is live if you go there, so those of you that want access to the slides, you can have them. So it turns out that there are 26 mouse phenotypes that have been built from the 310 genes that were communicated 2 years earlier. And 13 of those have been confirmed with regularly expressed phenotypes. And then we also have about 4 different phenotypes that have legacy. So these are well annotated. They actually have a 20 major phenotype categories, and it's really an intriguing project because in each of these you can drill down and look at the associations both at the morphological cellular level. They look at skeletal changes, weight increase, etc. And some of these proteins are G protein coupled receptors or ion channels, and those that I highlighted are GPR116, BA1, and this potassium channel, they have the most significant alterations. So it's quite an intriguing way to validate targets. Some thoughts on directions. We currently track drug targeting disease, and we are not doing very well with tissue presence, but we're improving that as we speak. And even more difficult, we do not know where the drugs go in tissues because there is not enough data there. I mean, volume of distribution is a way to measure that, but that's a very crude way to measure it. It's also difficult to ascertain the clinical outcome, whether it's beneficial or side effects, so we would have to go through the literature for each particular target, as I mentioned that for the mode of action. We have increased the target granularity, so we have this TCLIN plus, and we think that most likely the targets that are in TCLIN but not TCLIN plus will be associated either with side effects or with drug repurposing projects. So that would be a pool of targets for drug repurposing. Also potentially TCAN. And then we have this method which I skipped where we evaluate novelty and importance. And there's a system which we call TNEX that would allow target mining. There's a strong ongoing effort to integrate links and other omics data, which is done here at Mount Sinai. And we're working on doing another list of prioritized targets for IMDC. And then we're also working with Russ Altman and Tian Liu at Stanford to try to evaluate druggability for those targets that are in T-dark and T-gray, which have a 3D structure. So this is a algorithmic system that we're developing together. So a definite challenge is how to effectively communicate the prioritized targets. So let's say we have a list of 32 targets, as I mentioned earlier. Who cares? And potentially we could have anywhere between 10 and 1,000 targets to be prioritized. And just to give you an example, so on the Innocentive website, Boehringer Ingelheim is awarding a one-time 200,000 award for someone that will come up with a new target for asthma. And if you think that the asthma market is worth $20 billion a year, buying a target for $200,000 is like peanuts. Okay? So IP and who does what, I mean, all those things are really a challenge. So I'm going to stop here, and I'd be happy to answer any questions on that.